Uh, Good evening, everyone. Let's begin today's session. Uh, Tomorrow also today's session basically will kind of we'll have questions, and then we go back to the concepts, and then try to answer those questions. And tomorrow again, there will be an extra tutorial session if you guys want to join. Anyways, all these recordings are available. So tomorrow again, whatever questions we haven't covered or anything, then we'll try to do some extra questions as well. So let's begin today's. We'll start sharing this screen. All right. So the first mammal, Dolly, was cloned in which of the following? Cloned in during which time? So Dolly was the first cloned mammal. Okay, and it was done basically around in 1997. Was the late, like, nearing 2000s. So actually in 1997, Ian Wilmot and his colleagues at Roslin Institute in Edinburgh successfully cloned a sheep called Dolly. Okay, and Dolly was the first clone mammal. And Wilmot and his colleagues transplanted a nucleus from a mammary gland of this sheep into the enucleated, okay, or like enucleated into the enucleated egg of a Scottish blackface eel. The nucleus egg combination was then stimulated, like fused with electricity, so that these two can fuse and then stimulate cell division. So this is the donor egg taken from this sheep, and an enucleated uh, this was taken and then fused to promote cell division. And the new cell then divided, and it was then placed in the uterus of the blackface ewe to develop. Okay, here. And then Dolly was born months later, and Dolly was shown to be genetically identical to the fin dorsate mammary cells and not to the blackface ewe. Okay, um, like this, the Dolly that was cloned, like the uh, born, uh, it was the clone mammal was actually similar to this particular sheep, not to the sheep inside which the uh, fused cell was implanted. Okay, so this actually clearly demonstrated that she was a successful clone, and actually this was done after around 276 attempts before uh, like, Dolly was finally born. Okay, so anyways, the answer to the question, so it was cloned in 1997, 20th century. Option C. The prokaryotic ribosomes are composed of a larger dash and a smaller dash subunit. It is 50S and 30S. And for eukaryotic ribosomes, it's 60S and 40S. So the 30S and 50S subunits are they combine to form the 70S ribosomes. Basically, we talked about sedimentation coefficients a little. Okay, so sedimentation coefficients like 50 and 30 aren't really adding up to form 80. It combines to form a 70S ribosome due to differences in how the sedimentation coefficients work in ultracentrifugation. So these values are not really additive, but this sedimentation coefficients depend on the shape, size, and density of the particles. So when we talk about uh, uh, like prokaryotic ribosomes or eukaryotic ribosomes, these are basically ribonucleoprotein complexes. Okay. So if we talk about the 50S or in general, the prokaryotic ribosomes, you have 50s ribosomal subunit and 30s ribosomal subunit. Okay. Now, ribosomal subunits are made up of ribosomal proteins and ribosomal RNA. Okay. So, the 50s ribosomal subunit, the larger prokaryotic subunit, consists of 23s ribosomal RNA and 5s ribosomal RNA. And 23s ribosomal RNA is approximately 29, like 2.9 kb long, and this 5s ribosomal RNA is around 120 uh, base pairs, okay, long. And there are ribosomal proteins. There are 31 ribosomal proteins in the larger subunit of bacteria, okay, bacterial ribosome. Now, in the smaller subunit, the 30th subunit, you have like it has 16 as ribosomal RNA, and which is around 1.5 kb long, and the 30th ribosomal subunit is composed of 21 ribosomal proteins. These are the individual subunits, and when it assembles and associates to form the 70th ribosomes. And in prokaryotes, you have like 28 is 5.8 is ribosomal RNA. Okay. You have 5s as well. And there are around 50 ribosomal proteins in the larger 60s subunit. We are talking about eukaryotic ribosomes here. And the smaller subunit, which is 40s, it has around 1.9 kb long venous ribosomal RNA and 33 ribosomal proteins. And these two, 60s and 40s, associate to form the 80s ribosome. Okay? Fully assembled 80s ribosome. All right. So the gram-negative bacterial cell walls are not able to retain which of the following. Basically, this refers to gram staining. Now, if we look at gram-positive and gram-negative bacteria, here, like in the cell membrane and cell wall, this is the cell membrane, and the cell wall has a like thicker peptidoglycan layer in gram-positive bacteria, which also has different kinds of constituents. Teichoic acid, there are definitely surface proteins, then lipoteichoic acid. So you see, this is a thicker peptidoglycan layer. In gram-negative bacteria, what they have is the cell membrane, and the peptidoglycan layer is little thinner. Then there are lipoproteins, and then there's an outer like lipopolysaccharide rich outer membrane. Okay, this is in gram-negative bacteria. So due to the differing composition of their cell walls, outer layer, basically some some of them can actually retain the gram stain that they are stained with, and some retains the counter stain. Okay. So what happens here is um, when when we are staining the both types of cells, gram positive as well as gram negative, so both cell walls can affix the dye. All right. The dye complex is trapped in the wall in gram-positive bacteria. Okay, we saw that it's a thicker peptidoglycan layer. However, because of the outer lipopolysaccharide layer in gram-negative bacteria, um, what also happens is both cell walls are affixing the dye. Then, when we are adding iodine, there's the dye complex is trapped in the walls of gram-positive bacteria, but there's no effect of iodine on gram-negative bacterial cell walls. Okay? And then what happens is the crystals remain in the cell wall, okay? But because there are lipopolysaccharides, when you actually wash it with alcohol, then the outer membrane gets weakened and the cell wall loses the dye in case of gram-negative bacteria. And then when you add a counter stain, so what happens here is when there's counter stain, these still retain the gram negative stain, that crystal violet stain. Okay, but the counter stain is taken up, and the counter stain is given, like we see that counter stained cells for in case of gram negative bacteria. Okay, because the due to alcohol wash, the whatever uh, dye, the initial crystal violet dye, purple dye that was taken up is lost by the cell wall. So these are the steps that happens. First, apply crystal violet, you stain the cells. Then you actually add iodine, which is a mordant which affixes the stain on the cells, and then you wash it off, which leads to decolorization of uh, these uh, these cells. And then you add this counter stain, which is usually saffronin, and then you can distinguish easily between gram positive ones, which retains the stain, the purple stain, and gram negative ones, which actually loses the dye, the first dye, and then gets stained with the counter stain. So the gram-negative bacterial cell walls are not able to retain which of the following. The initial stain that was used. So option will be option C, crystal violet. Which of the following is the cofactor of the enzyme hexokinase? Now this question relates to the uh, glycolytic pathway. Also hexokinase we saw it there. And the cofactor that is required for this enzyme is divalent magnesium address. Cofactors are something that helps the enzyme to perform its function. So again, if we just brush up glycolytic pathway, it is divided into two phases, mainly broadly. One is the preparatory phase, which requires energy, and then there are processes in the payoff phase which releases energy, okay, in the form of ATP in glycolytic pathway. All right. So, like, so you have glucose, which is a 6 carbon moiety. So preparatory phases basically phosphorylation of glucose and its conversion to glyceraldehyde 3-phosphate. And glucose is a 6 carbon moiety, which is finally broken down to 3 carbon glyceraldehyde 3-phosphate, and another compound 3 carbon compound is formed, which is DHA. So first you have, this is the first priming reaction. Glucose is converted to glucose 6-phosphate by uh, where 1-phosphate from ATP is used. Okay. And the enzyme for this conversion is hexokinase. Kinase is any kind of enzyme that phosphorylates, is a kinase. And because it is phosphorylating a 6 carbon moiety, it is a hexokinase here. And then glucose 6-phosphate, 6 carbon moiety, gets converted to fructose 6-phosphate. So this is also a 6 carbon moiety, just that there is an isomerization going on here. All right. So the enzyme that catalyzes this reaction, the conversion of glucose 6-phosphate to fructose 6-phosphate, is phosphohexose isomerase. Right. And then you have then fructose 6-phosphate. Another phosphate is added on the fructose 6-phosphate. Still, this is still a 6 carbon moiety, just there's an addition of phosphate group here. All right. So if it requires a phosphate, again, it uses an ATP for taking up one phosphate group from there. And because there's phosphorylation going on, the enzyme in play is kinase, which is phosphofructokinase. This actually is catalyzing the conversion of fructose 6-phosphate to fructose 1,6-bisphosphate. And then finally you have, now this is the step where the 6 carbon moiety is getting cleaved into 2 3 carbon molecules. Okay. So this is fructose 1,6-bisphosphate gets converted to glyceraldehyde 3-phosphate and DHAP or dihydroxyacetone phosphate. Okay. Cleavage of 6 carbon sugar phosphate to 3 carbon sugar phosphates. One is glyceraldehyde 3-phosphate and another is DHAP. And this conversion is brought about, this catalysis is brought about by the enzyme aldolase. All right. And then glyceraldehyde 3-phosphate. So preparatory in the preparatory phase, 2 ATP molecules were used. Okay, because there were 2 phosphorylation events going on. Now what happens here is in payoff phase, you have oxidative conversion of the three carbon compounds to pyruvate and the coupled formation of ATP and NADH. So glyceraldehyde 3-phosphate is oxidized and phosphorylated. And then this is an oxidation and phosphorylation step where two inorganic phosphates to NAD plus come into play, and then it forms like this 1,3-bis phosphoglycerate, again a 3 carbon molecule, and 2 NAD plus comes in and NADH is given off. Okay, NADH is produced. And then, yeah, this conversion is brought about by glyceraldehyde 3-phosphate dehydrogenase. And then 1,3-bis phosphoglycerate is converted to 3-phosphoglycerate, where like here two phosphate were added, but then now one of the phosphates is given off, thereby forming 3-phosphoglycerate. Okay, you see there are two phosphate groups attached here. One of them has been given off, and that along with the ADP, it forms ATP molecule. So energy producing reaction is going on here, and this reaction is catalyzed by phosphoglycerate kinase. Okay. And then you have the conversion of 3-phosphoglycerate to 2-phosphoglycerate. Like from this position, the phosphate gets shifted here, which is brought about by phosphoglycerate mutase enzyme. And then from 2-phosphoglycerate, have phosphoenol pyruvate being formed, and you see an addition of double bond here because H2O gets released, and this conversion is brought about by enolase. And then finally phosphoenol pyruvate is converted to pyruvate. Okay, so the phosphate group here gets released and it is taken up by ADP molecules to give off it to form ATP, and hence this is the second ATP forming reaction, this substrate level phosphorylation. Okay, and this conversion is brought about by pyruvate kinase, right? So the previous question was about hexokinase, the very first enzyme in the conversion of glucose to O6-phosphate. Now select the incorrect pair among the following: gel filtration chromatography, surface area, affinity chromatography, affinity to specific substrates, ion exchange chromatography, molecular mass, and hydrophobic interaction chromatography, hydrophobic patches. Basically, what is the basis of separation in all these different chromatographic techniques? So, gel filtration chromatography definitely it uses the overall hydrodynamics surface area of the uh, sample in question. Whatever is the constituent of the sample, whichever is the larger molecules, okay, those do not have access to the pores, they easily pass through, and then the larger molecules are the ones to elute up first, as compared to the smaller ones, which can easily get inside the pore of the matrix in the gel filtration column, and hence those smaller molecules elute out later. Okay. Then you have affinity chromatography, okay, where the affinity between different groups are explored, where suppose you have like histidine tagged protein, histidine tagged molecule is there, and your matrix has divalent nickel ions. Okay. So there's the affinity between nickel and histidine is exploited. Okay. And then first you let the histidine tagged protein bind to your uh, Ni like nickel ion conjugated matrix, and then what you do is you pass imidazole, which has play around with the concentration of imidazole, so that first initially you let your proteins bind to it under low imidazole concentration, and then you increase the imidazole concentration so that now there are more number of molecules to bind to nickel ions, and your protein, your histidine tagged protein gets washed off. So affinity chromatography, I mean, this is one of the examples of affinity chromatography. There are various other examples. So we read about it in the previous tutorial sessions. So affinity chromatography, affinity to specific substrates. Ion exchange chromatography basically actually exploits the charge, the overall charge on your sample, on the constituent molecules of your sample. Okay. So it's not really dependent on molecular mass. And hydrophobic interaction chromatography definitely depends on, explores the hydrophobic patches under different kinds of salt concentration. Okay. So the incorrect pair is definitely ion exchange chromatography, which doesn't really depend on the doesn't really exploit the differences in molecular mass of the sample. Rather, it exploits whatever is the charge of the constituent molecules of your sample of interest in a particular pH. Because increase in pH and decrease in pH actually interferes or actually kind of changes the overall charge on your protein sample. Now, in a PCR reaction, polymerase chain reaction, the denature of the double-stranded DNA is done by heating the reaction mixture at which of the following temperatures. So definitely first the DNA has to be denatured so that you have the single strands onto which your primers will anneal, and then eventually there will be uh, replication. I mean, like amplification of all those uh, individual templates. So it is at a high temperature, which is usually 95 degrees Celsius. And many times there's 98 degrees Celsius as well. People use in some cases. And 95 to 98 degrees Celsius initial denaturation step is used during PCR. Okay. So, but out of these options, the correct one will be option B, 95. Now let's look at it briefly. So there's initial denaturation, which leads to melting of double-stranded DNA to single strands, all right? And then what happens is, first there is initial denaturation, and then denaturation initiates these cycles finally, mostly 25 to 40 cycles. Then you bring down the temperatures so that you have the specific gene-specific primers, which can anneal at their specific sequence. And then there's a thermostable DNA polymerase, which elongates the primers, okay, which is the elongation step, the first where primers are uh, going to their binding to their complementary regions is annealing. Then a thermostable DNA polymerase elongates the primers because there are DNTPs, I mean nucleotides that you are adding in the solution as well in the reaction mixture, from where it takes up the polymerase takes up those nucleotides and keeps on adding one by one, which is the elongation step. And then after elongation, the target DNA strands are duplicated. Okay. So overall, due to 25 to 40 cycles of these reactions go on, and repetition of these three essential steps are the basis for exponential DNA amplification. Finally, you have from suppose a few molecules of your gene of interest to millions of copies of your gene of interest. Okay. Exponential application amplification allows the accumulation of the specific PCR product. Correct? Now, when we talk about the primers, we have to be very careful while designing the primers because that will eventually, I mean, there are other things to take care of, but unless we have the specific properly designed primers, our gene of interest might not get amplified in a proper way and it might not have the exact sequence that we want. Right? So primer length matters. Oligonucleotides, usually the length of oligonucleotides are 18 to 24 bases, which is long enough for adequate specificity and short enough for primers to bind easily to the template at the annealing temperature. Now, primer melting temperature, which is the TM. Primers with melting temperatures in the range of 52 to 58 degrees Celsius generally gives the best results, and the GC content of the sequence gives a fair indication of the primer TM. And the two primers should be prepared, like the forward and reverse primers, should be prepared in such a way that the difference in their melting temperatures should not be more than 2 degrees Celsius, like 2 to 5 degrees Celsius. Otherwise, it will actually result in a poor annealing efficiency, because what will happen is, at this particular temperature, suppose one, if there's a lot of difference between the annealing temperatures of your forward primer and your reverse primer, what might happen is, suppose your reverse primer will bind the template strand to its complementary template strand. However, the forward primer won't be really specific. So the overall amplification will be affected. Okay. Primer annealing temperature. Too high, of course, a too high annealing temperature will produce insufficient primer template hybridization, because they will tend to get denatured, which will result in a low PCR product yield, while too low annealing temperature will lead to non-specific PCR products caused by a high number of base pair mismatches. And then, of course, we have to be wary of the GC content. If there are a lot of GCs, because it's a tighter binding there, right? There are three hydrogen bonds there. So the number of G's and C's in the primer should be in between 40 to 60%, because if there's a very high GC content, then it will be a tighter binding of the primers. So eventually when the cycle goes on, it will be tighter binding will not let the primers like really go off for the next denaturation cycle, okay, next cycle. And if the GC content is too low, again it might be non-specific. Okay. Then there's something called as GC clamp. Also, if the GC content is higher, the GC forms a stronger bond than AT, right? And the number of GC content at the 3' end of the primer should not be more than 3. Otherwise, it will result in non-specific tight binding at regions where GC are abundant. Also, um, we should be wary of the dimer formation, like self-complementarity between the forward and reverse primers. A primer dimer, so if the forward and reverse primers are binding to themselves rather than the complementary strand, definitely the overall efficiency of the amplification, PCR amplification, will reduce. Okay. A primer dimer is a structure forming a double strand-like structure which is formed by intermolecular interactions between the two primers. If interaction is formed between the homologous or same sense primers, it is called a self-dimer, whereas if interaction is formed between two different primers, it is called a cross-dimer. Okay. So whether it's a self-dimer or a cross-dimer, it is a problem, because eventually it will affect its binding to the complementary template strand, and hence finally the amplification will be affected. When we talk about the repeats and runs, repeats are consecutive occurrence of dinucleotides, whereas runs are continuous stretch of a single nucleotide. Okay. And a maximum number of repeats, and generally accepted maximum number of repeats and runs is 4 dinucleotide uh, repeats, consecutive occurrence of dinucleotides, and 4 base pairs respectively. Primer template homology. Primers should be designed in such a way that there should be no homology within the template other than the target site, right? If there's any other site, like suppose you have a complete 3 kb with, okay, 3 kb plasmid, and then your gene of interest that you want to amplify out of that is barely 1 kb long, right? Or maybe 500 bases long. So if that is the scenario, then if your design primers have uh, complementarity or some other target sites as well, complementary to some other target site other than your gene of interest, which is 500 base pairs, like outside of those 500 base pairs, if there are complementarity between your forward and reverse primers, then what will happen is there will be non-specific amplification as well. Okay? So we have to be careful about that. So in general, for a primer length of less than 14 nucleotides, the TM is usually measured as in degrees Celsius. Is 4 into number of G's and C's in the primer plus 2 into number of A's and T's in the primer. And for a primer length of more than 13 nucleotides, so TM is approximately um, 65 degrees Celsius plus 41 into number of G's and C's and divided by N, where N is the number of nucleotides in the primer. Okay. And now, the optimal agarose gel percentage typically used to view an amplicon of 30 kb is what? Now, when we are talking about the agarose gel percentage, so agarose is forming pores. Okay. Now, the percentage of agarose is actually regulated based on or decide the percentage based on what is the size of the DNA or like nucleic acid fragment we wish to visualize. If it's a smaller fragment, definitely if the pore sizes in the gel are a lot bigger, then the smaller sized fragments won't be nicely resolved. Okay. However, if the larger pore sizes are really large in an agarose gel, what will happen is the bigger fragments, bigger nucleic acid fragments can actually pass through it easily. However, if the pore sizes are too small, then the really smaller fragments, like suppose 50 bases long or 100 bases long, those ones can easily pass through the pores, okay, and pores of the agarose gel matrix, and then form like can be nicely resolved. However, like really large fragments like a 30 kb fragment, it won't be able to pass through it properly. To pass through the small sized agarose gel pores properly. So usually we modulate these uh, percentage like amount of agarose. So basically, when we talk of when we say that there's 2% agarose gel, that means that in 100 ml of TAE buffer or in general TAE buffer or TBE buffer, whichever buffer you wish to use, uh, in 100 ml of buffer, 2 grams of agarose. Okay. Basically, W by V, this is the percentage, this is a W by V percentage. Okay. Now for 30 kb is too long a fragment, the amplicon. So it will need a lesser percentage of agarose. Okay. So it will be option C, 0.5%. Alright. Now, which of the following restriction enzyme cuts the DNA within its restriction site? So there are different kinds of restriction enzyme which can cut at the within the site of its recognition or few base pairs like 400 or 1000 base pairs away from its recognition site. Alright, so we did look at few of the examples in the earlier tutorial sessions. So the answer is type 2. So what happens here is this is a type 1 and it cleaves like type 1 cleaves at sites remote from the restriction site, requires both ATP and SAM, and then also these do have three types of uh, like functions. One is the restriction digestion activity, one is the recognition or DNA binding domain, S subunit, and then there's M subunit, which has a methyl transferase activity. Okay. And type 2, the recognition of type 2 enzymes is 4 to 8 nucleotide long and its recognition site is 4 to 8 nucleotides long and it cuts the DNA within that site. Okay. And then this is why yes. Can you be a little louder? Oh, previous slide. Okay. So, okay. This one. So shall I move on? Oh, Uh, Actually, you will also have the recordings available. Okay. So yeah, again, type 2 restriction enzymes are also composed of three subunits, the M, R, and S, where S is the DNA binding domain, R is the restriction, which actually shows the restriction dilation activity, and M has the methyl transferase. And then again, if we talk about type 2 enzyme, type 2 actually can also have different types. Okay, like type 2B, which again is a multimer. It can cleave DNA on both sides of the recognition to cut out the recognition sites. So few examples are BCG1 and BPL1. Then there's type 2E, which can cleave DNA after the interaction with two copies of the recognition sequence. The first recognition sequence, it is actually the target for cleavage, and second has the allosteric effector. Then there's 2F, which can interact with two copies of the recognition sequence again, but one cleaves, but in this case, the 2F actually cleaves both the sequences at the same time. Then there's 2G, which is a single subunit enzyme and requires SAM as a cofactor. Type 2M recognizes and cuts methylated DNA. And there's 2S, which is a dimer, cleaves DNA at a defined specific distance from their non-palindromic recognition site. There's type 2T, which has two different subunits and can recognize palindromic sequences. Okay. Then there's something called type 3 restriction enzymes, which has two separate non-palindromic sequences arranged inversely oriented, and the cutting site, restriction site, is nearly 20 to 30 bases away or nucleotides away from the recognition site. And again, type 3 enzyme is composed of two subunits, RES and MOD. MOD is required for modification, mostly methylation of the host DNA, whereas RES subunit cuts the unmethylated DNA. When we are talking about enzymes, then especially for the daily experimental or laboratory purposes, we need to use, suppose for cloning, we have to, in a regular day-to-day basis, we have to use enzymes. Now, when we are using enzymes, we have to keep the amount, keep a track of the amount of enzyme that needs to be added in a reaction mix. Okay? So enzyme unit actually quantifies an enzyme's catalytic activity, and it is denoted by U, which is micromole. Basically, 1 U is micromole per minute, or if you want to define it, it's the amount of enzyme that catalyzes the conversion of 1 micromole of substrate per minute under the given conditions, okay, under the specified assay conditions. And these conditions, what are the assay conditions, which include mostly include factors like temperature, the pH of the buffer in which the reaction is set, and the substrate concentration, which yield the maximal substrate conversion rate for that particular enzyme. And enzyme activity is expressed in units per milliliter, okay, or as nanomoles per minute per milliliter. Okay. This is what is U, like nanomoles per minute is the U that we looked at, and then per milliliter gives enzyme activity. Basically, it's relating to the concentration and varying. It actually varies depending on the enzyme's role in metabolic pathways, DNA replication, gene expression, and different other activities, like different other kinds of reactions or metabolic processes. There's something called turnover number, represented by K-cat. Like cat will be a subscript here, not a like like K, where subscript it will be C8. Represents the maximum number of substrate molecules an enzyme's active site can convert to product per second when this particular enzyme is actually completely saturated with its substrate. Okay, this is the turnover number. And there's something called star activity as well. We should be careful of this uh, activity for in case of enzymes, where under non-standard reaction conditions, some restriction enzymes can actually digest nucleotides like DNA sequences similar to but not really identical to their defined recognition sequence. And factors contributing to star activity can include glycerol concentration, like the glycerol, basically glycerol in any kind of reaction mix or buffers actually makes it viscous. So if the reaction mix or the reaction conditions are like really too viscous, then the enzyme that is not really an optimum condition for the enzyme to function. Okay. Then there's buffer choice. Suppose the pH or the salts are not really optimum for the function of the enzyme that you are adding in that reaction. Reaction time. Many times, suppose you wish to when you're digesting a nucleotide sequence, many times if you keep it for a prolonged time, it starts cutting at non-specific sites. So when you really want, uh, suppose a linear DNA wants to cut it, uh, really digest it using restriction enzymes. So ideally, you should be seeing two strands after digestion, right? But what might happen if you are extending the incubation times, reaction time, what might happen is you will see a smear or some other non-specific products in the reaction when you run it on agarose gel. Okay? So reaction time also matters, and the presence of organic solvents, which might actually affect the activity of the restriction enzymes. So star activity basically leads to cleavage at non-canonical recognition sites or even sometimes complete loss of specificity. Okay. Now, alkaline phosphatase is used to do which of the following functions? Okay. Alkaline, if the name suggests that this is something that is used to remove the phosphate groups. Okay. Phosphatase means it is removing the phosphate groups. So the answer will be option D. Is removing the free phosphate groups from the vector. Now, which of the following is not really a good characteristic, like a preferable characteristic for a good vector? High copy number. We definitely want a high copy number because we want the recombinant plasmid, our recombinant plasmid inside which like the vector basically, inside which we have cloned our gene of interest to really, divide and produce any number of recombinant plasmids. We want our recombinant plasmids in a good amount of quantity, like good quantity, good concentration. It has to be a low molecular weight because if it's too high, like it's really a heavy molecule that gets inside the cells, it can be toxic to the cells. Okay, that will be a burden. Anyways, introducing uh, plasmids, like external plasmids, recombinant vectors inside cells is a little burdening to the cells, but so that's why it's preferable to keep it uh, like use low molecular weight vectors. There should be multiple cloning sites with unique restriction sites because that is where you will cut off your, like digest your vector, and then with the same restriction digestion of your insert, you will insert it using sticky end ligation, right? Okay. But definitely you want to have a selectable marker. So you really, if the vector has gone inside your cells, the recombinant vector has gone inside your cells, then you need to have a marker to say that, okay, these are the cells that have actually taken up those recombinant vectors, okay, or recombinant plasmids. So you need to have some kind of change in the cells or some kind of, suppose antibiotic resistance, some kind of lack of uh, any phenotype or addition of any phenotype. So that needs to be there. Otherwise, you won't really be able to deduce that your recombinant plasmid has been taken up by the cells. So we really want a phenotypic change inside the host cell so as to detect the same, so as to detect the taking up of the recombinant plasmid. So if which is not a criteria, it's option C, no phenotypic change to the host cells. Okay. Now, which of the following compounds is used to prepare competent E. coli cells? So, mostly divalent salts are used, like salts of divalent cations are used. So, the answer will be option C, calcium chloride. So, basically, the most conventional and most regularly used, one of the most regularly used ways of transformation chemical is chemical treatment, where you give the treatment of calcium salts and magnesium salts, treat the cells, and then you play around with the temperature. Okay. So the calcium treatment and temperature treatment actually interferes with the um, like the composition or pore like movement of the lipids and proteins on the outer layer of the cells, like component cells, and hence it helps in taking up the recombinant DNA. Okay. And also the treatment with calcium ions as well as magnesium ions actually helps in uh, the like either negating the charges or masking the charges on the outer layer of the competent cells, and also helps in the absorption of the DNA molecule. Okay. That is the temperature imbalances and CaCl2 help deal with the barrier to take up foreign DNA, like charge repulsions and pore sizes. Now, spheroplast transformation is used to transform which of the following types of cells. Now, in spheroplast transformation, it's actually used for yeast cells. What happens there is yeasts have a cell wall, right? So that has to be digested. So that is actually mostly the enzyme used is zymolase, which helps in digesting the outer layer of yeast cells. And then actually incubate it at certain temperatures. Then you add a carrier molecule that conjugated to your DNA, and the vector overall can be taken up and inside the cells again grown under specific conditions like optimum temperature conditions for an optimum period of time, and then you will finally have your yeast cells with the with your required gene of interest. So basically, spheroplasts are very fragile for osmotic shock, but are competent to take up free DNA at high rate. So what another like polyethyl glycol peptide is used to facilitate the deposition of plasmid and carrier DNA on the cell wall for easy uptake by the cells. So this is to directly answer the question. Spheroplast transformation is used to transform the yeast cells. Option A. Now, a vector with a restriction site located within URA3 was what? Oh, yeah, all right. So, a vector with a restriction site located within URA3 was used to insert a gene of interest. After transformation into E. coli, the cells were plated on solid agar containing 5-fluoroorotic acid. But no colonies were observed after overnight incubation. Which of the following inferences is incorrect? So this is something based on complementation of mutation. Okay. So what actually is happening here is a mutant host strain can be used to screen the plasmid containing a missing gene or a like knocked out gene, and the transformant will only grow if the gene product from a clone will complement the function. Okay. So in general, genes that take part in the metabolic pathway or any biosynthetic pathway of the cells inside which we like the cells that are routinely used for taking up the recombinant DNA, that is actually used. So three important requirement is the host strain has to be deficient in that particular gene. If the gene belongs to any biosynthetic pathway, then the mutant host in this case are called as auxotroph as host dependent on the gene product or final product of the pathway as a supplement in the media for growth. Okay. And a defined media with the missing nutrient and a vector containing the gene to supply the gene product to complement that particular mutation. Okay, so there are yeast actually has 4 different genes like HIS3, U2, PRP1 and URA3 as a selectable marker. Okay. So here actually what happens is if like no colonies are observed after overnight incubation, what might have happened is OMP decarboxylase was non-functional, which couldn't really complement the like couldn't really complement the functioning of URA3, okay, and conversion of 5-fluoroorotic acid. Okay, so So what is happening here is one is again when we are talking about complementation of mutation, there can be either positive selection or a negative selection. Okay, the URA3 one is a negative selection. So, but in case of positive selection, what happens is the host strain doesn't grow on the media which lacks the functional gene, but only the host cells which are transformed with the recombinant clone are able to supply the gene product which is required for those cells to grow in the media, which is lacking a certain kind of nutrient. And there's negative selection, where a chemical compound is added to the media, which will be converted to cytotoxic agent in the presence of a gene product. And as a result, it doesn't really allow the growth of wild type cells. But the host strain transformed with the recombinant clone has non-functional gene product and grows in the presence of the compound in the media. All right. So the URA3 here actually encodes for OMP decarboxylase, which helps in the conversion of 5-fluoroorotic acid to a toxic product, okay, which is fluorodeoxyuridine, and hence which is toxic to the cells, and then no colonies are observed. However, if you actually insert any kind of gene here, then this will be this won't be able to encode, this will be a non-functional uh, uh, like this will encode for a non-functional product, and hence this decarboxylase won't be able to act on, like won't be able to catalyze this conversion, and hence you will be able to see colonies. Okay, so that is what is happening there. If you are seeing no colonies there, that means this carboxylase is non-functional and it couldn't really convert uh, like 5-fluoroorotic acid to fluorodeoxyuridine. This is the toxic product. Okay. This is the same thing. URA3 codes for OMP decarboxylase, the same thing which has been shown here in the picture. Now, a protein in the native state exists as a tetramer. The molecular weight of the protein is found to be 48 kDa in denaturing conditions, and the native molecular weight will be uh, in denaturing conditions, it is 48 kDa. So it will basically be 4 times uh, the molecular weight because it is existing as a tetramer. Okay, so option will be option C, 192 kDa. The electrophoretic mobility of a protein is directly proportional to which of the following? Uh, Basically, the electric field, the portability of an ion is not entirely set in stone by the molecular size, shape, and charge, and the temperature during separation. It is actually consistent under the characterized electrophoretic conditions. Okay, and these ones we read about it in general. So another thing, uh, there's a opposite viscous drag on the charged moiety when it moves in an uh, like in an electrophoretic medium. So the relative mobility is directly related to the charge by radius of the molecule. Suppose for a globular protein in question, the radius of the molecule is related to the molecular mass of the macromolecule. Okay. So the electrophoretic mobility of a protein is directly proportional to the charge on it. Okay. And it is actually, the mobility is inversely proportional to the size as well, because the greater the size, the larger the size, it will take longer to move through the uh, matrix or the gel. pH of the resolving gel in STS page is uh, option C, 8.8. The glowfish and the stacking gel is 6.8. Okay. The glowfish is a genetically modified fish that expresses a fluorescent protein that can perform photosynthesis, which can crossbreed between species, shows chemiluminescence. It actually, glowfishes are something which expresses a fluorescent protein. Now, there is something like chemiluminescence and fluorescence. There's a difference in there. When we talk about chemiluminescence is something which is the emission of light as a result of a chemical reaction, and fluorescence is actually emission of light from a substance that has absorbed energy previously. Okay. like we shine a light on the fluorophore tagged molecule, okay, and then it absorbs some energy from the uh, light, and then it emits light from a substance, like as in the fluorophore emits light that initially had absorbed some amount of energy. Like in chemiluminescence, what happens is a chemical reaction which forms low energy product from high energy reactants. However, in fluorescence, what happens is electrons are excited due to absorption of external energy. And definitely what you really are observing in chemiluminescence is after the completion of a chemical reaction. And fluorescence you observe as soon as you remove the source of the radiation or the source of the light. And as soon as you remove it, something, whatever energy the fluorophore was able to absorb, then then it emits light at a corresponding wavelength. In the toxoid class of vaccines, the dash is used for immunization of the subject. So, if we are talking about toxoid class of vaccine, the inactivated toxic compounds are actually used as antigens for immunizing the organism in question. So there can be different types of vaccines. One can be live attenuated vaccines, which are mostly in cases of tuberculosis, oral polio vaccine, measles, yellow fever, rotavirus. Then there's inactivated, like killed antigens can be introduced to stir up an immune reaction, uh, which can be pertussis or inactivated poliovirus. And you can actually maybe introduce a part or subunit of the toxin in question, and this is used in cases of Haemophilus influenza type B, pneumococcal, hepatitis B. Okay, and there's toxoid class of vaccines which are actually inactivated toxins, like in case of diphtheria and tetanus. There are RNA-based vaccines as well, which are definitely non-replicating. In vivo, there can be in vivo self-replicating as well, and in vivo dendritic cell, but non-replicating. Okay, but the question is about toxoid class of vaccines, which are actually inactivated toxins. So option D, inactivated toxic compounds. You can actually also, if you wish to, all these slides will be is uploaded in the drive, so you can go through it if you wish to, like really read more about it. All right, then that's it for today's session. So we can actually I have frozen. Anyways, we can meet if you guys are free. You can join tomorrow. Otherwise, there will be the link, YouTube link for the recording. All right. Thank you so much for joining, and if possible, we'll see you tomorrow as well. So, any questions? What we have discussed in the tutorial sessions. Tomorrow, we'll try to discuss questions different from these tutorial sessions. Okay. So, yeah, yeah, no, thank you for joining. Okay. So someone wants to join the call now. Yeah, can you be a little louder? Uh, my session is from 6 o'clock. I mean, in the evening. So that is there. But other sessions, I think I am not really aware of. So my session will be. Oh, I am not really aware of actually. Okay. So, all right. I will end the session for today. Thank you for joining.